音，这样听得到我讲。我说话有哈，那说话我听得到吗？你可以拿我的那个无线耳机，然后对着他讲话看看。你应该把调掉，然后耳机拿着就可以讲话，但是记得离电话远一点。
Hello，
我可以多讲点话吗？
你有听到我吗？
可是它是一个 hello 这样子的回音，你一直讲话，我调一下。
等一下哦，等我一下。哦，正错了。已经两个都打开哦。
好啊，
听得到吗？
你现在这个比较清楚，反正应该没
好，
问题，因为听起来有声音就好了。
好，那等一下回来再看一下。
同学，拜拜。
拜。那我关静音喽，拜。
Hello, Professor Scheltens.
Hello, hello,
How are you?
hello. Wait a minute. Yeah, I'm here.
I can hear you. It's okay.
Hi, good morning, good morning.
Morning. Okay. We will start a few minutes later.
Okay, fine. I have the, I can share my slides, I think, in a while.
Okay. Actually, you can share your slide right now.
Okay, I'll do. Wait a minute. Wait a minute. Oh, shit. Wait a minute, wait a minute. Okay. Good idea. Okay. I have to leave the meeting. I will be back in 1 second. Okay, good morning. Here I am again. And now I'm going to, let me see. Oh, wait a minute. Can you see this?
Yes, we can see that.
Can you see my slides?
Yes, we can see that.
Okay, good. I will put them on full screen. Wait a minute.
Mhm.
Can you see
Yeah,
it now?
right now it's okay. Very good.
Good, good. Okay, we'll just wait. Thank you for the invitation.
Yeah, it's our honor. You can give a talk here. Very good. Thank you.
Well, how is the situation in Taiwan? Is it okay with you?
Yeah, I think it's okay. Yeah, it's like a military crisis, but right now I think it's peaceful. Yeah.
Okay, okay. I hope it will stay peaceful for you. I like Taiwan a lot. I've been there a couple of times and it's a really beautiful country.
Thank you.
Beautiful people. Oops, wait a minute. So,
Professor Scheltens, can we start?
I'm fine. Yes, whenever you are, whenever you're
Okay,
ready.
thank you. Good afternoon in Taiwan and good morning in Amsterdam.
Yes.
I am Dr. Chaur-Jong Hu, the director of Dementia Center Shuang Hospital, Taipei Medical University. In this year, we are funded by National Health Research Institutes to conduct a continuous education program for our dementia physician and dementia care team members. We have the continuous program we named Frontier in Dementia Research Master Lectures. The master today is Professor Philip Scheltens from Alzheimer Center, Amsterdam, UMC, Netherlands. He founded the Alzheimer Center in 2000, and since this year, he also is the head of EQT Life Science Dementia Fund. Professor Scheltens is the first researcher to raise the MRI criteria for hippocampal atrophy of Alzheimer's disease. It's well known as medial temporal lobe atrophy score or Scheltens score. He also established Amsterdam Dementia Cohort. It currently is the largest collection of clinical and biomarker data. Based on the big biobank, he together with colleagues raised the new diagnosis research criteria for Alzheimer's disease based on the presence of clinical phenotype and the presence of a CSF biomarker and the molecular image in terms of ATN system of NIAAA research framework. The title of his talk today is The State of AD Field. Let's welcome Professor Scheltens.
Thank you very, very much, Professor Chaur-Jong. It's a real pleasure and a real honor to speak to you today on this important topic. And I know there are many, many, many topics that fall under the umbrella of the state of the Alzheimer field. So I have chosen a little bit to go in the direction of therapy, because I think that is the most important direction that we will have some developments in this year, actually, and also last year. But I'm happy to take also questions on the criteria or on biomarkers at the end of the session. You can ask whatever you want. And we will focus a little bit now on the developments in the therapeutic field, which relate a lot to the developments in the biomarker field in the past. So as said, I am indeed very much involved mainly with clinical trials nowadays. I'm PI of the studies with AC Immune, Fujifilm Toyama, UCB, and also studies with Vivoryon, Novo Nordisk, and Novartis. Since 2 years, I'm also involved in venture capitalist in sort of to really help companies to develop drugs by supporting them with some money and also with advice. So I'm now also an investor together with a large team to help companies develop drugs for the future. This is no secret to you, and I think the situation in Taiwan is probably the same as in the rest of the world, that the prevalence of dementia is increasing, and we know this for many, many years already, and this is not news. The problem with dementia, in contrast to other diseases, is that the burden for society is much bigger than it is for oncology or cardiovascular disease. And that is why dementia as a number of patients, the prevalence is so important. It has an enormous burden and costs associated with it. And that makes it actually the largest and the biggest healthcare challenge for the future. Now, you see that there are many targets on goal actually for dementia. For the sorry, I have to remove this, otherwise I don't see it. In the biotech industry to tackle Alzheimer's disease, and this has been going on for quite a long time, but not as long as for oncology, for instance. So I think the real therapeutic area in Alzheimer's disease has only started 20 years ago, really. And the main focus in the industry still is amyloid as the driver of Alzheimer's disease pathology, but also tau and also neurodegeneration. And as you know, these three elements, the A, T, and N, form the body and the sort of the main pathology of Alzheimer's disease, upon which we now have based the criteria and using the biomarkers. But you also see that, for instance, neuroinflammation, genetic factors, metabolic pathway, glucocorticoid and antioxidant and antibacterial and many, many others are also being used nowadays. And I will come back to that later. This is a nice slide of an article that we just published a couple of months ago together with some people from Amsterdam Neuroscience, the Clinical Trial Research Center, people from EQT Life Sciences. And we took a look at the drug development field in Alzheimer's disease in the preclinical phase. And as you can see here, it is still the main body is still formed by, in the preclinical space, by amyloid, and in the clinical phase, it's a bit less in amyloid. So preclinically, there are still a lot of drugs developed in the amyloid beta space, but also tau, preclinically more than clinically, actually. But as you can see, neurotransmitter receptors, inflammation is a big part here, both preclinically and clinically. And as you can see here, the other ones. This is a very interesting one, ApoE, lipids, and lipoprotein receptors. As you can see here, a lot is happening also in the preclinical phase. So the message of this slide is that the field is really moving forwards, not only focusing on amyloid and tau and neurodegeneration, but has a broader perspective on the pathology of Alzheimer's disease and how to treat it. And I think that is very, very wise. The failures of the past with bapineuzumab, with solanezumab, have all to do with the fact that we were only focusing on amyloid and didn't use the right drugs. So I'll come back to that later as well. So there is more to choose from, and there is a lot of activity in the field. So don't be pessimistic. We can be actually optimistic about the future because there are so many companies working and the targets are so pluriform, I would say. So this is how the situation is at the moment. It's actually just changed also a couple of weeks ago. This is the amyloid beta targeted drugs in phase 2 and 3 clinical trials. And what is important is that, in fact, all the studies nowadays are focusing on the very early stages of Alzheimer's disease. And that is only possible because we can use the biomarkers nowadays. So we can identify patients much earlier than we could years ago. Also, interestingly, there are a couple of studies focusing on familial Alzheimer's disease, but both of them, as you heard very recently, the crenezumab study from AC Immune and Genentech also failed to produce clinically meaningful outcome measures, although they had an effect on the biomarkers. So these red ones, they failed, but they're still important. The two on the top, the gantenerumab study and the BAN2401 or lecanemab, they will present their data on the phase 3 results this year. End of this year at CTAD, we will have the readout of gantenerumab, and just a month before we will have the readout of the lecanemab studies. And I think those are two very, very important, very large studies with huge number of patients, very well conducted, biomarker-based patient enrollment, and clinically meaningful outcome measures, namely the CDR and some of the others. So, important this year, keep in mind, and perhaps we should do this webinar again in a year or so, because then the situation may have changed completely. A few others here, important, I will come back to that one, donanemab. This will read out next year, early next year, also a monoclonal antibody directed against A-beta. Now, what are there are differences between all these antibodies, and I will show you. Oh, wait a minute. This is another one. This is very important and interesting because I was also involved in the very early start of this particular small molecule. This small molecule targets a very toxic form of A-beta, the pyroglu A-beta. This pyroglu A-beta is crucial in the formation of synaptotoxic oligomers. It does not occur in normal aging, and that's important. So amyloid beta plaques, A-beta 42 plaques, you see in normal aging as well, but pyroglu A-beta is not happening in normal aging. It is typical and very specific for Alzheimer's disease. Now, we know that oligomers already before they form plaques have an impact on synaptic activity, and hence pyroglu A-beta toxicity may cause memory problems. Now, pyroglu A-beta is formed by an enzyme, which is glutamyl cyclase, which is this one. And so in conjunction with the appearance of A-beta 42 and A-beta 40, this specific enzyme also forms these pyroglu A-beta oligomers. Now, this particular drug, PQ912 or varoglutamstat, inhibits the glutamyl cyclase. And why is this interesting? Because it also works on A-beta, a specific form of A-beta, but it is an oral taken formula, so it's a pill, and it is a small molecule that enters the brain. And that's very important. So no infusions, no subcutaneous administration, just a pill. So where are we with this? This is the phase 2A study. It was just a small study to look at the safety, the tolerability, and a little bit also and the efficacy. And what we saw is that there were some elements that actually supported the mode of action towards synaptic changes, because we had influence of this particular drug, PQ912 or varoglutamstat, on neurogranin, which is a specific synaptic marker, as you can see, and YKL40, which is an inflammation marker, but also a cognitive test, a one-back test, that we saw sort of significant results here, and also on EEG, which is a reflection of the synaptic function, we saw an effect. And this was only a 3-month study. This was only a study to look for subtle effects to base the phase 2B study on. So no definite proof at all, but the drug was safe and well tolerable, and we then moved on with a little delay to formulate a phase 2B strategy, and this is done both in Europe and this is also done in the US. The European study is the VIVAT study. It's a phase 2B study in early patients, Minimental 2030, who are amyloid positive. And the primary endpoint is cognitive function. This is the neuropsychological test battery. And we have also biomarkers and, of course, EEG to be measured. I am the PI of this study until the end of this year. The duration of the study will maximally be 96 weeks. There was a titration period to go to the maximally tolerated dose, and we just released a couple of weeks ago, there was a press release that actually the dose titration worked much better and there were less side effects than in the phase 2A study. So the dose is now set to 2 times 300 milligrams. The same is happening in the US with a little bit delay. They started later. They will have a 24-week study. They will have sort of a dose-finding study, actually, with cognitive function by the ADNI group and the EEG. We'll sort of see how that plays out, but this will be a little bit later. as you can see here, this will not happen because the patient recruitment is far behind. They just included a few patients, so this needs to be shifted to the right, while we, on the other hand, will stop recruitment at the end of this year, in September actually, and the results will be known in the second half of 2023. Keep this in mind because this is a very important situation. Also, because of Simcere, which is a Chinese pharmaceutical company, has licensed this drug particularly to test it also in Chinese patients. I'm not sure whether this will also happen in Taiwan, given the situation, but just for you to know that there is a Chinese company that actually took an interest in this particular compound. Now, how do all these sort of monoclonal antibodies and the directions sort of differ from each other? And is there another player? Yes, there is. So here we have aducanumab from Biogen, we have donanemab, and we have Eisai. So these two, and the gantenerumab is not on the slide because it was too much. Basically, the important thing is that these are all IV infusions. 10 milligram per kilogram was the effective dose in the Biogen study. This is 700 milligram monthly, and this is 10 milligram per kilogram also twice per month, so bi-weekly. This is all tested in early AD, actually, and as you know, there was an effect in all of these studies on the biomarkers, CSF p-tau, and also imaging biomarkers like amyloid PET, as you can see, and there is, and there is an effect on all these markers in all the studies, and especially in the Eisai study, this is not on the slide here, there was 80% reduction of amyloid burden using lecanemab. Same actually with the Lilly compound. Now, this is interesting. There is another compound, which is from Alzheimer, which is the ALZ801, also a small molecule, also targeting oligomeric function. And this is an ongoing phase 3 study, but only in ApoE4 homozygotes. Because this is a very old drug. This was tested already many, many years ago in 80 patients with no effect, but in a post hoc study, they showed that the effect was present, but only in ApoE4 homozygous. So this is a sort of precision medicine approach. If it works only in ApoE4 carriers. And as you know, ApoE4 homozygotes are roughly 16% of the AD population, 1/6. They have, and I can tell you this because this was in the biomarker study that they are conducting, which is ongoing now. They reported the results at ADPD and also at AIC. They showed a 30% reduction on p-tau in CSF. They have no brain edema, they have no ARIA at all. Well, these therapies all have side effects, as you know. So these drugs will probably enter the market much earlier. My bet is that lecanemab has the best chance to get a clinically meaningful results, but beware, these are infusions bi-weekly. They're very burdensome for the patient, and they also have a little bit of risk for ARIA, of course, although less, as you can see here in the carriers and the non-carriers, less than in aducanumab and also less than in donanemab. These were the results from Lilly, and this makes actually the circle very nice because donanemab is also targeting pyroglu A-beta, the same as I showed you with varoglutamstat. But varoglutamstat is a small molecule, while donanemab is a monoclonal antibody. But they both target the same pyroglu A-beta species, which is very toxic. This was a phase 2 study that was published in the New England Journal just last year. All patients that were included were amyloid positive and tau positive, so they fit the ATN criteria. There was a very nice clinical endpoint that was positive, which is a combined cognitive and also functional scale, the IADRS, and they had a huge reduction of amyloid PET plaque burden. Now, as I said, the phase 3 study uses almost the same design, but they have also every patient amyloid PET positive and tau positive. So we'll be curious to see the results in the beginning of next year. So, but again, monoclonal antibody, IV infusions, but a very smart design with a very good target, which is pyroglu A-beta, which makes it different from the normal A-beta that happens in normal aging. Now, as I showed you earlier, there are things happening beyond amyloid, aging of the cell, synaptic function, mitochondrial dysfunction, genetic factors, lysosomal dysfunction, inflammation. And there's also a lot of variability in how to approach the disease. So we have seen small molecules, we have seen the monoclonal antibodies, but there are antisense therapies in development. There is stem cell therapy in development. There is gene therapy in development. There's active vaccination in development. And there's also RNA interference. So there are many, many more shots on goal. I will show you the shots on tau here. This is a bit of a problematic field because in tau, which is, as you know, intracellular, so how to attack phosphotau and phosphorylated tau, which is in the cell. This has been a challenge. So you can see in the tau targets that some target the N-terminus, some target the midbrain, mid area of tau, some target the tau mRNA, which is the Ionis, the antisense approach. Some target the monomeric or fibrillary tau, mid-region, the C-terminus, etc., etc. Well, some of them have failed. Of course, LMTX and also the AbbVie drug and also a Genentech Roche drug, monoclonal antibody. Also, the ones that are still, this has now also failed. I have to make this also red. The Lilly compound is still in the race. This is a very promising compound, but they have some issues with starting up the trial. This is an active vaccine, actually, against the truncated, misfolded form of tau. I think that's probably the best target, but there are some issues, so they haven't started yet, the trial. This is an AC Immune and Johnson study together. This is a vaccine and a monoclonal antibody developed at the same time. So the field for tau therapy is much younger nowadays, but it is present and it is sort of approaching phase 2. One word on the antisense approach. We know this from other disease areas as well. It is quite simple, actually. It is the antisense approach delivered by intrathecal sort of administration to bind actually the messenger RNA, in this case of tau. So if you bind the antisense oligomer to the messenger RNA, in this term anti-tau, there is no tau protein anymore synthesized. So it's actually a real cut down of the amount of tau in the brain. And already at biomarker studies that have been done with this specific specific ISO from what's it called? Now I block on the word. It's actually Ionis, actually, and they showed that they have a very, very good p-tau sort of effect. Now the clinical results will be presented next year. So interestingly, but again, invasive therapy. This is intrathecal injection, and we have done the study also, and it is burdensome for the patients, but they have 5 LPs and 5 times an injection of the ASO. Now, importantly and interestingly, ApoE4 has been known as the most important risk factor for Alzheimer's disease for many, many years. In 1993, Alan Roses discovered that ApoE4 carriers had a larger, bigger risk for Alzheimer's disease. So you would think that ApoE would be a very, very good target for therapy. If you can lower the amount of ApoE4 in the peripheral system and lower it to ApoE3 or ApoE2, you actually de-risk the patients. And this is now finally being tried by some of the companies that target actually specific pathobiology that is occurring in ApoE4 carriers or on the ApoE4 receptor, or to compensate the loss of ApoE function, because ApoE4 has less of a function in the cholesterol metabolism in the brain than ApoE3 or ApoE2, or enhancing the neuroprotective function of ApoE2, which is, as you know, a protective factor. So all of these sort of efforts are ongoing in targeting ApoE4 in ApoE4 carriers as a risk factor. If you do this early enough, you would actually de-risk the patients for developing Alzheimer's disease. The same goes for anti-inflammatory approaches, the microglia response. You know that the microglia responds to amyloid presence. When there are as amyloid toxicity, you see inflammation happening. TREM2 is involved in that process, and many others like TGF-B and NROS actually, and many others. And this is also TREM2 particularly is a very interesting target for drug development at the moment. And some companies are working on that as well, but that's early days. So, you know, Jeff Cummings publishes every year this huge figure actually, and this is not to be read by you, you can try, but this is just to illustrate again that the field is really very active and very lively. Phase 1, phase 2, phase 3. There are many, many drugs, there is many approaches, small molecule, disease modification, small molecule, symptomatic small molecule, and disease-modifying biologics, for instance, the monoclonal antibodies, as you can see here listed. So many shots on goal, as I said earlier, and you can see this here. This is the drug development pipeline that is summarized by Jeff Cummings. There's actually a new version just came out at ADPD. I will modify the slides soon. So how does this play out? I think what I find very interesting is that we of course want to modify the disease by targeting all these elements that I mentioned earlier, but still patients have symptoms and you need to treat those symptoms as well. And actually we have very little to treat the patients with. So we have, of course, the cholinesterase inhibitors, but they have a very modest effects and not long-lasting effects. So we need also to have symptomatic drugs to be developed. And that is a bit disappointing. So we have only a few % of the agents that are in phase 2 at the moment are symptomatic. So cognitive enhancers or neuropsychiatric symptoms, agitation, depression, psychosis, etc. That's a bit disappointing, I think. And this is what we need to pay attention much more, because the patients suffer from this much more, and they will not, and I will repeat, they will not notice any effect if you do a disease modification therapy, because you can't compare yourself to where you would have been without the therapy. So this always will be need for symptomatic treatment, even if you have a disease modifying therapy. And here again, there are very many disease modifying therapies in phase 2. And here again, you see the armamentarium of everything that is happening. This is phase 3. This is a bit of a higher number, neuropsychiatric symptoms and cognitive enhancers, but still the majority is disease modifying therapies. Now, many of you and also many colleagues in the field are a bit skeptic because they say, well, we have had so many failures in the past, so it can't go right. I mean, there are too many failures, so the field has to do better. And I think what Jeff Cummings and Howard Feldman and myself have tried to summarize is, why is this happening? Why has it happened in the past? And what can we do better? And we formulated this as the 5 rights of precision drug development for Alzheimer's disease. And this may sound like an open door to you. This is very sort of logic thinking, but it hasn't been applied to all the previous trials, actually. So if you talk about the right target, it is, of course, it's amyloid, it's tau, it's neuroinflammation, but it has to be druggable. It has to be a real target that's meaningful for Alzheimer's disease pathology. So backed by genetic mechanisms, for instance, or by clinical mechanisms. The target needs to be meaningful for the disease that you are targeting, in this case, Alzheimer's disease. And I think that's already a very important start. And I've shown you all the targets that people are working on. Then, of course, the right drug, which you think, yes, that is logic, but is it the right drug? Does it have dose response relationship? If you increase the dose, does it also increase the effect, or vice versa? Does it enter the brain, for instance? Those very sort of elementary questions have to be asked before you start the trial, because if you do the trial and you have not established that the drug targets the brain or enters the brain actually, then you have a failed trial in the end. The right biomarker, of course, this goes to, first of all, patient selection. So do they fulfill the A and the T criteria? But also, can you use your biomarkers for target engagement and target selection, and probably also outcome measures? I mentioned already a few times that some of the monoclonal antibodies had an effect on A-beta on PET, but also p-tau in plasma and CSF. These are examples of target engagement and target effect using biomarkers. Now, the right participants, I think many of the studies in the past have used patients who were too late in the disease, too far away, and too much damage had already been done. So the general feeling now is that you have to target very early. And how early is early? Perhaps in patients who have no symptoms yet. But that becomes, of course, very difficult, because how do you select these patients when you don't have symptoms? I think the sort of the compromise in between is to select patients who have at least complaints, subjective memory complaints, and are biomarker positive. That is the earliest selection you can use for patient participation, and this is also happening at the moment. And then, of course, the number 5 right is the right trial. Now, again, this may seem obvious to you, but a lot of trials in the past have not been powered well enough to see an effect on a clinical readout. And I think the clinical readout is probably the most important thing. And I think I can spend a whole hour talking on clinical endpoints, which are the right ones and which are the wrong ones. And I think we have been stuck for far too long with the ADAS-Cog and with other types of endpoints that were relevant in the past, but not anymore. So you have to do the right trial with the right amount of patients and the right endpoints. And you have to think really, really very hard which endpoint matters to the mode of action of my drug that I'm testing. So for a symptomatic drug, you can probably use still the ADAS, but for a disease modification drug, you really need more sensitive endpoints, cognitive endpoints, but also functional endpoints, to show that over a longer period of time, patients actually decline when they are not on drug, and they may improve when they are on drug. And especially I would pitch that the functional outcomes, like the ADCS scale or the Amsterdam ADL scale, are extremely important to use, because they measure very subtle, important functional changes that matter to patients nowadays. So the patient of today is different than the patient for 10 years ago, because patients do mind if they can't handle the telephone, they can't make electronic payments anymore on the computer. That sort of subtle instrumental activities of daily living are an important functional endpoint that is being used now in trials. So the 5 rights you can use as a sort of cookbook when you develop your own clinical trial. So, my conclusion from this short overview is that I'm actually very positive. I think the future looks brighter than the past. There are 2 important readouts this year, a 3rd one next year. There are many shots on goal currently tested by small and a little bit larger biotech companies. They are for amyloid, but also more importantly, a lot of non-amyloid therapies are being tested. The amyloid hypothesis is by far not dead, as some people try to have you believe because of the failures of some of the trials. No, it is not dead, but it hasn't been tested properly. And I think the trials that are now ongoing have a better design, have a better drug and better targets. We will never, ever have one single therapy to cure Alzheimer's disease. We always will need a combination of therapies. At least amyloid and tau and neurodegeneration, synaptic function, inflammation, perhaps genetic modification as well. I think that will be important. Like in oncology, like in HIV/AIDS, you will have to have more than one therapy. My belief now, since I'm working much more in the biotech sort of sphere, is that innovation comes from biotech and not big pharma. Big pharma has been sleeping for a long time. They have not done the right job, and the real innovation comes from the smaller companies. We have seen this in COVID with the development of the vaccines. Happily, I think that the 5 rights are implemented widely, and as I said in the beginning, I think the future looks much brighter than the past. And with this, I will just hand it over to the chair, and I'm really very happy to answer all the questions that you would like to ask.
Okay, thank you, Professor Scheltens. A wonderful talk. He updated the drug development of Alzheimer's disease for us. Now, Professor Scheltens' talk is open for discussion or comment. Okay, I have the first one question. We read the report about the oligomeric Alzheimer amyloid beta, A beta star 56 from the study of University of Minnesota. What do you think about the research?
Yeah, so that's a very important question, and I'm happy that you asked this. I think it was also very well covered by Alts Forum. This was a paper that was published in 2006 by one particular group at the University of Minnesota, as you mentioned, and it has not been replicated by other groups. That's number one. Number two is that it was 16 years ago, and there has not been research based on this particular hypothesis of the existence of a particular subform of oligomeric A beta star 56 that has produced sort of trials or new drugs. So in fact, actually this particular publication wasn't even noted so much, and it wasn't even influential, and it doesn't challenge the overall amyloid hypothesis. It doesn't challenge the existence of oligomeric A beta, because everybody recognized this has been replicated by many groups all over the world, but this specific subspecies has not been found by other groups. Now, it may be that the issues, that the figures were fabricated, that it was an invention, and if that is the case and it is proven, that is of course very harmful for science in general, but the finding itself has not been influential for the Alzheimer field in total, and it will not change our thinking about amyloid beta or oligomeric A beta species.
Okay, thank you. The second question is, I noticed your VVET trial, you use the EEG as a biomarker. Can you explain the role of EEG in the Alzheimer's disease?
Yeah, that's a very good question. So I'm a real fan of using EEG as a biomarker. We know that it is of limited diagnostic value. We see slowing of the EEG signal from the alpha waves until the theta waves, of course. We see general slowing happening in Alzheimer's disease, relatively a little bit later in the disease process, but it has been proven to be very efficient in showing an effect of those drugs that are targeting synaptic function, because EEG itself, and especially if you do analysis of the EEG signals, you can derive a sort of marker of connectivity from the EEG signals. And you know, the brain is all about connections. Synaptic function is important for connectivity, and EEG is a measure of connectivity. So if you have drugs targeting synaptic function, like varoglutamstat and sort of some others actually that target oligomeric function, EEG may be used as a measure of whether this particular drug actually changes the ratio between the normal signal in the alpha band and the slowing of the signal in the theta band. And we have shown in the phase 2A study that actually that was the case. And luckily, many other groups in the world are now using EEG as an outcome measure. And I think that has to be proven still. It will never be the single outcome measure. It's only supportive, also to show that the drug is changing something in the brain. So it's, again, proof of target engagement.
Okay, thank you. There are other questions. One from Dr. Li. What's your opinion about the difference between two biomarkers for neurodegeneration, NFL and GFAP? Which one do you think is more suitable as a biomarker outcome for a clinical trial?
Very good question. Very good question. I'm not sure whether somebody of you was at AAIC, but there was a lot of buzz around neurofilament and GFAP. So neurofilament light is a direct neuronal marker, and it can be measured in plasma and it can be measured in CSF. The nice thing about the NFL is that it is completely A-specific, but it is very sensitive. So it can show you changes in neuronal function and neuronal loss, actually, in diseases like ALS, multiple sclerosis, frontotemporal dementia, and Alzheimer's disease. So it's not specific for one disease, but it is sensitive to the changes. And even in multiple sclerosis, for instance, you see an immediate drop in neurofilament light once you start therapy, and when you stop the therapy, it goes up again. It has been shown that neurofilament light has a very good signal function when changes are happening in the brain that make patients symptomatic. For instance, in the pre-symptomatic gene carriers in the PS1 population, you see as soon as the neurofilament starts to change, that is the point in time where patients start to develop symptoms as a matter of fact, as a signal of neuronal loss. So a very good marker to use for neurodegenerative process over time. And that's been used all across the board in neurodegeneration. Now, GFAP is a bit of a younger marker. We have done some research on it. It can be measured also in plasma, also in CSF, and it's a real sort of astrocytic marker. So it's a little bit more specific for Alzheimer's disease. And the combination of NFL and GFAP in Alzheimer's disease yields a very high diagnostic accuracy. And if you combine it, for instance, with A-beta 4042 and phospho-tau and ApoE4, you get 100% characterization of your population. And as a diagnostic tool, I think for the future, the four elements, A-beta, phospho-tau, NFL, and GFAP, may be used as a combination of markers to identify those patients who have the disease and already are in the stage where changes are beginning to happen in the neuronal and astrocytic sphere.
Okay, thank you. 永轩，你可以直接问，打开麦克风直接问。Dr. Lin Yongxuan will ask her questions.
Professor Scheltens, I have a question. Will the antisense oligonucleotide approach harm the microtubule stability if no tau protein were synthesized, as you mentioned earlier?
Sorry, yeah. So what was the question? Sorry, I couldn't understand the question. Sorry. Can you do again?
Okay. Yeah. Will the antisense oligonucleotide approach harm the microtubule stability is that no tau protein were synthesized at all.
Yeah, that's the key question. That's exactly true. So if you completely block the formation of tau, you have, of course, tau is a very useful protein, and it's used, and the microtubules are extremely important. So that has been the criticism for this particular therapy as well. So far, I can only say that in the phase 2 study that has just been ended and the results are analyzed, no particular side effects have been noted. The changes in p-tau were interesting, but not that huge, so it wasn't completely gone to a normal level. So I don't think it blocks the sort of complete synthesization of tau. So I'm less worried, but it's a very, very, very good remark, and we have to see more results for this ongoing phase 2 study before we can make the final judgment.
Okay.
Okay, thank you.
Thank you. I have another question. We see the more and more like blood-based biomarkers for diagnosis. Do you think in the future we still need amyloid PET for diagnosis or outcomes survey?
That's a good question. Again, if you were at AAIC, it was the main topic actually at the conference is plasma phospho-tau 217. And the interesting thing about plasma 217 p-tau is that it's actually, although it's the name says p-tau, it's actually a very good amyloid marker because it's only positive when amyloid is also present. So I think in the future, I foresee that we will use these plasma biomarkers perhaps in a combination, A-beta 4042 and plasma p-tau 217 in combination as a very early marker. If they are abnormal and you want to treat patients, for instance, with a monoclonal antibody, I think you will need at least amyloid PET confirmation. But when the trials will have been done without amyloid PET as an entry criterion, will not need it anymore. But now I see the role of p-tau in plasma as a pre-screening and a screening and a diagnostic first step before you decide to do amyloid PET, before you do your therapy. Without therapy, I think p-tau would be brilliant to use for that as a diagnostic support up and above your clinical evaluation of the patient without amyloid PET.
Okay, thank you. The other question is how to explain the more atrophy of hippocampus in ESAI study, not a good marker.
Yeah, that's been troubling many people. So that's actually very difficult to explain, and I hope it's not. It's perhaps it was only in the phase 2. I haven't seen it in other studies. We know, of course, that from other earlier monoclonal antibody studies, but actually also in the active vaccine study earlier on, in the early 2000s, we saw that removal of amyloid may actually result in more atrophy. Now you may argue there's not that many amyloid, not that much amyloid in the hippocampus, but there is a lot of A-beta oligomer changes in hippocampus, and since lecanemab is actually targeting the protofibrils / oligomers, it may actually mean that by removing those, you get some volume loss. But as long as the patients don't deteriorate but actually do better, it apparently has no functional correlate, this volume loss. So it may be a bit less, how you say, swelling because of the protofibrils. It's really, you can see I don't have the answer, and we'll have to see what happens in phase 3.
Okay. 魏如你可以自己问，打开麦克风自己问。Okay, but Dr. Lee will ask his question. Can you?
Yeah, yes. Yeah. Thank you, Dr. Scheltens. I have a question about the blood-based p-tau biomarker, and which platform do you suggest that we used to check the blood-based p-tau biomarker?
So To my knowledge, the best results are, of course, used by the mass spec, but mass spec is a very sort of elaborate, expensive, and can only be used in one laboratory. I think the next stage, and what we use also, is the Simoa platform. And the Simoa platform is widely distributed in many hospitals, has the ability to detect very small signals, and also has the ability to perform multi-assay assessments at the same time. So we use the Simoa platform to look at A-beta 4042, p-tau, GFAP, and neurofilament light all at the same time.
Okay.
Thank you.
Is there other questions? I saw the Professor Dai is here. Do you have any comment or questions?
Oh, thank you. Please.
Hello.
Thank you for your very nice lecture. I personally learned a lot, and I'm very surprised, but very happy you are so optimistic for the successful clinical trial in the future. Thank you, and looking forward to see you in Taiwan very soon.
Yes, please. I would love to come. I'm optimistic by nature, so maybe that is my fault. But I don't think we have to really let go the optimism, despite all the things that are happening. I mean, as you mentioned, the A-beta, sort of the oligomers 56 is a blow for science, but not for Alzheimer's disease. We shouldn't let ourselves distract from the goal that we have. I think aducanumab is a very good example of a very good drug, actually, and reaching its target very, very well. But the trials were, of course, hampered by the fact that they were incomplete. But again, at AAIC, there was an FDA official who really made the case very clearly why they approved aducanumab. And actually, he said we wanted to go for full approval because we were very convinced that the data showed an effect clinically, and they went for accelerated approval as a sort of compromise. So I really think we have to be patient, we have to do our work properly, we have to do the proper trials, and in the end we will see some success, and then the field will change and everybody will be more optimistic, not only me.
Okay, thank you.
There is a question, what is the best outcome in terms of an endpoint be set in AD trial? If it is a cognitive performance, what would you suggest if any specific form is preferred?
Yeah, I think there is no general answer to this question. I think every trial has to choose the endpoint that is relevant to the mode of action. So, for instance, plaque removal by the antibodies is in a sense not immediately leading to a specific cognitive change. So it is, I think, worthwhile to use a more global scale like the CDR to look for effects over a longer period of time. When you're using small molecules and you're targeting a specific oligomeric species like in the varoglutamstat, and you directly hit a synaptic function, then I think memory tests, sort of sensitive memory tests, are the best outcome. In general, I think, and that's a European view, we always have to look also for functional changes, because I really think that our patients do not mind very much whether memory is a little bit less worse than it was before, but what they can do in daily life is much more important. So I think the development of composite scales using cognition and function are, I think, very promising for the future. And there are a few that have been developed that show a great response to therapy, because you increase also the power if you combine elements from two specific domains into one.
Professor Wang Shujun is here. You can open your microphone. Professor Wang, please.
Yeah, thank you very much for your talk, and I learned much from you. And another question is, how early do you consider is early enough for these clinical trials to be successful?
Yeah, that's a
I know
good
this is a
question.
difficult question. Yeah.
Yeah, it's a difficult question, and I've tried to answer it already a little bit. I mean, there is the trade-off, there is the balance. The earlier you go, the longer the trials have to be. Because if you go for, and that is the lesson that we have learned from the PS1 carrier studies, if you include patients who have the gene and they have no symptoms at all, they may be 5 to 10 years from the estimated age of onset of symptoms. So you have to do very, very long trials. So in order to mitigate the length of the trials and to look for an effect in say a period of 18 months or 36 months, I think the best choice for me would be to go for patients who have subjective complaints. So already they have complaints and they are biomarker positive, because those patients will come to the memory clinic. You don't need to screen the population. You can just use your memory clinic population, but they have to be biomarker positive, of course. And I think that's a sort of compromise in between the ideal situation that you have patients without any symptoms but with the disease. But how to find these patients? You cannot go screening in the population, even not with plasma p-tau. That's absolutely not feasible. So you have to do a compromise. And I think using memory clinic population with subjective complaints is the best compromise.
Thank you.
Thank you. Any other question or comment? Okay, I think we have almost run out our time. Today we are thanking Professor Scheltens for his wonderful talk, and he answered a lot of questions from the audience. Yeah. In the future, I look forward your visiting to Taipei, Taiwan again. So we close this talk here. Thank you.
I'm very
Thank you
much
so much.
looking forward. I'm looking forward, and I'm very much supportive of Taiwan, and I love your country, so I'd love to come to Taipei again. It's been many, many years ago, actually. Yeah, so if we have some more news next year, it's a good time to meet, probably.
Okay, thank you. Thank you,
Good.
all of the audience. Goodbye. Bye.
Bye.
Thank you.
Bye-bye. Bye-bye.
Bye-bye.
Bye.
Enjoy the weekend.
Thank you.
You too.
Thank you.